So Dr. Florence Mehl. Florence is a computational biologist at the Swiss Institute of Bioinformatics in the Vital IT group in Lausanne, in Switzerland. She has a master's degree in analytical chemistry and a PhD in organic chemistry. She is an expert in chemometrics, particularly in the integration of multi-omics data, including in human health research projects. She has also been an active member and treasurer of the RFMF for many years. And Florence, I'll hand it over to you. And I think we have to switch off the mic of the other participant because it will be a nightmare otherwise.
Hi everyone, good afternoon. Thanks a lot to the organizer for inviting us, me and my colleagues. So today I will talk about the multiomics data integration, especially based on the chemometrics method. We will be free of the SIB working in Lausanne, Josefina, Vanu, and myself. We are working in Lausanne at the SIB, which is a network of scientists and also a non-profit and independent institution that is involved in many different bioinformatics related activities, from developing and maintaining open databases and softwares, providing support to research in academia, hospitals, or industry for data management, analysis, or training, and also coordination of national and international projects. So we are facing the data explosion in our everyday work as modern technologies produce massive data sets. From the hypothesis-driven approaches, where you start with a scientific hypothesis, design experiments, and produce focused data, we are now often following the data-driven approach, where you acquired a lot of untargeted data with help of bioinformatics and statistics, and then you mine the data to draw hypotheses. To follow this path, 3 different steps are necessary. Data production, as you may know, you prepare your sample, acquire your data, extract the signal, clean the data, normalize, annotate, etc. Then you analyze your data to explore or classify, do pattern recognition with a lot of different statistical methods, or AI methods, or whatever methods you like in bioinformatics. And then last but not least, you need to interpret the results of this analysis and put the data into biological context and then validate. So during this talk, I will show you one way of doing data analysis, the integration of multiple blocks or tables of data. These blocks could be of different data sources. For example, they could come from or be acquired from cell, from tissue, from organism. It's part of systems biology, but they could be also from a different stage of a process, dose, toxicity, disease progression, or different analytical techniques that gives heterogeneous data. You can have different separative or spectral methods, for example. So the nature of data of the different block could be heterogeneous. The data block can be all measured on the same scales or not on the same scales. They could be quantitative, they could be qualitative, ordinal. So, but why do we need to integrate them? So we can combine all those data sources to gain an extending understanding of complex systems. We can use the complementarity of data to obtain more complete or new information, but we can also use the redundancy of data to increase confidence. For example, integrating LCMS and NMR, for example. We can also combine different data sources that are complementary to better predict an outcome. But we can also integrate the data to compare data sources, for example, to rank data sources according to the information they provide, or to select one block that provide more information or more relevant information to the phenomenon you are studying. So how could we integrate all those multiple blocks of data? There's a lot of different methods, but we can classify them generally in three different families. The network-based method, which tries to integrate networks of different networks of data, for example, similarity network fusion or molecular networks. You can also apply deep learning methods, all the family of neural network, the autoencoders, the generative AI, etc. And the matrix factorization methods, for example, the multiple factor analysis, or the MOFA in the Bayesian family, or COM-DIM consensus OPLS, which will be the focus of this talk today. The methods based on components that are, it's another name for matrix factorization methods. Why are they interesting? So they are interesting, they are well suited for omics data because they can manage high dimensionality data with correlated variables, also with large biological variability. And there are projection methods that are used to analyze those high dimensionality data sets. We can, with those projection method, we can visualize the samples distribution. We can understand the relationship between the variable, between them, and also how they behave in the different samples. So we can also find, for example, unsuspected relationship, and we can summarize the data with a small number of factors, which is really advantage of this type of method. In this family, we can and the others also, we have to divide the analysis between unsupervised and supervised analysis. The unsupervised analysis give you a way to explore the data. You are looking for structure and pattern, but with no a priori knowledge about the observation. So you really want to see how the data are, and if you see a signal that could be related to something you know about your data. In the supervised analysis, it's a predictive data analysis. You know a response, which can be, for example, groups or biological outcome, and you want to put emphasis on this response and see how the data could be related to this response and could help to predict this response.
Tu veux venir voir parce que j'ai un message d'erreur que je ne.
Ok, mais j'essaie de suivre un truc.
Supervised analysis, you may know the principal component analysis, which is really like the mother of all this family of methods. The PCA aim to compute the first component, that is the maximum variance from the origin. So you compute different components that are orthogonal the one to the other, and each of one try to summarize a part of the variance. The first one is summarizing the major part of variance, then the second one is summarizing the variance remaining after the removing of the first. And with that, you can end summarizing your original data, your matrix of the original data, with scores and loadings that are, the scores are the sample, the coordinate of the samples in the new space of component that were computed, and the loadings are the weights of the variable in this space. So this is the unsupervised analysis, and for the supervised analysis, you may know also the partial least square. And in this, with this model, the aim is to best explain the variance in your original data and to compute the component that best explain the variance in the response. So what you want to do is to use your original data to predict a response that could be groups or a continuous response, for example. You may also know the orthogonal projection on latent structure, which is almost like the partial least square analysis, but here you want to compute a predictive component that is really summarizing the variance that is related to the response, and all the other components that are called orthogonal component are summarizing other source of variation. So the variation in the orthogonal components are not related, information not related to the response you want to focus on. All those methods are performed on matrices. So 2D matrices, you have one data table with observations and variable. In multi-block, we are working with collection of matrices, meaning that you have different data table with observation and variables. So how to integrate or to fuse all those data? You can follow different strategy to integrate these different blocks of data. High level, which means that you integrate the results or the interpretation of model computed for each block. For example, you can do a PLS model for your different blocks and interpret each of the model, do the interpretation until the end, having new biological insight for each of the model, and then try to integrate this new information. The mid-level is made computing a latent variable for each data block, and then integrate those variables computed separately into a new model. In the method we will see today, we apply a low-level data fusion strategy, which means that we integrate the data, the matrices of data. I won't say raw because they were already processed, but you integrate really the matrices of data and not pre-computed latent variable. As you may know, the limitation of the high and mid-level data integration strategy are the following. So the results may be contradictory or heterogeneous. It's really difficult to integrate the results, the interpretation of the different models that you computed on the different data tables. It can be really tedious, and all the results can be inconclusive. You have also no insight into the link between the measurements. So between the different tables, it's really difficult to make links. And the individual model may lead to substantial loss of biological information because you cannot relate them. So with the low-level data fusion, where you integrate directly your different matrices of data, you can think global because you are building a compromise that accounts for all data, setting adequate weights between the blocks. And also locally, you want to maximize the link between the data block under specific criterion, which can be very various and needs to be chosen carefully to answer your question. But this enables to find the relevant information. You can see then the role, the importance of each data table to focus on common and specific trends and to make links, obviously mathematical links between variables of different nature. So here in this low-level data fusion, you need to think about the data structure you are facing. So either you can integrate vertically if you have different samples, for example, but the same variables that were measured. You can also have the same individual measure with the same variables, but for example, in different times. You can have also L shapes like that, which are more difficult to address. And here we will focus on this horizontal data fusion with the samples, the same samples in the different data tables, and the different blocks of variables. So to do this horizontal data integration, you can use a whole forest of methods. So as you can see here, in this image is coming from this really good book. You have a summary of all the different data, and you see that you have a large choice of data applicable. They are all working with this horizontal data structure where the observation mode, so the sample, are shared between the different data tables, but the variable mode, so the columns, are really specific to each data table. We have in the omics generally, and those methods are well suited for, we have many more variables than observations. And the goals are the same as for single block data analysis. We want to describe, we want to classify, discriminate, or predict the data with the data. Here in the horizontal multi-block analysis, so as I said, the observation mode is shared. So this is really important. If you want to apply the methods I will show you today, you need to have the same observations in the different blocks. You can do unsupervised analysis to explore your data, describe your data, look for structure and patterns, and you can find links between variables in a single block and also links between variables from the different blocks. With the supervised analysis, you can predict a response of block Y, which could be one of them. You can do connection between one or many blocks, and some blocks are dependent. They are, and some other will be independent as the response. The methods showed today as PCA are factorization, matrix factorization methods. So factor analysis can be applied as before, either to one block or to many blocks of initial variables. So each component, each computed component, it will be related to one block and is connected to all the components related from the other blocks. And they are related to a global component. The block components should verify those two properties. They should explain well their own block, and they are correlated with other components from the other blocks. The multi-block model build component has a compromise, so explaining between blocks, so the different latent variables should explain variation between the different data blocks and also inside the different data blocks. And you can, adjusting the weight and the algorithm, you can favor explaining more the within block variation or the between block variation. For the unsupervised multi-block analysis, this analysis can help generate hypotheses from the data blocks, undirected links also, and all blocks are treated in the same way, meaning that you can exchange the different blocks. The sequence of blocks have no meaning, no incidence on the model. And here we will focus on common variation and specific variation. And we need to have fairness between blocks to be able to compute the model and to gain insight of the results. So I will show you one of the methods that exists, which is called ComDim for common dimensions, or CCSWA for common components and specific weight analysis. You have all the different data blocks as inputs, and then you will compute, or the method will compute those square matrices that have the same size, which is the size of the number of samples. And then those matrices you do a scalar product with these different matrices. So each of them will have a weight of 1 to start, and then this weight will be iteratively adjusted. So the block weighting will be iteratively adjusted. So I will show you a small application of this method, which is part of a study on type 2 diabetes, where we had 3 strains of mice fed with 2 diets at 3 different time points, and a lot of different samples collected from different tissues at different time points. And we also had biochemical outcome. And we wanted to investigate the key organs that control glucose usage and production with those mice with different genetic background and sensitivity to a high-fat diet that induce metabolic stress. So we had transcriptomics and lipidomics analysis. So here you see that we have plenty of different data sets with really different sizes, and we've decided as a first step to go through a dimensionality reduction step in order to improve even more the fairness between the block, and gain also easiness in the interpretation. We apply then the modeling, the unsupervised modeling with a R package, which is called the MBA analysis. And for interpretation, it's like for PCA, you will have scores, which are the projection of samples, the loading, which are the contribution of the variable to the model. And here, a new thing that is specific to multi-block modeling, which are the saliences, which is the percentage of variance explained in each block by each dimension. And if we look at the score plot, you see that it's really like a PCA. You will color them. the model have no a priori knowledge, so the colors showed here are put afterwards, and you can retrieve or not your experimental design. Here we can see the different strains and the different diets. So we clearly see that our experiments had an effect, and that this effect is well shown also by all those different data sets, the transcriptomic data and also the lipidomics data. Those are the two first dimensions, and you can see how many explained variants is summarized in each dimension. So then you can see which block is more related to which source of variance. But then so here we described the data. We see that all the different data sets have information related to the biological phenomenon, but we wanted to understand how the mice were regulated their fasting glycemia. So then we jump to a supervised multi-block analysis, where the blocks are no longer exchangeable, because you have a block of response here, and you cannot exchange the block of data with the block of response. So here, same as before, you will compute latent component, latent variable from the different block, and you want to regress those latent variable on the Y response. We follow the consensus OPLS algorithm with all those heterogeneous data as before, with the association matrices, the square association matrices, a weighted sum matrix, and here the weights are iteratively computed, adjusted to give more weight to the block which is more explaining the answer. But we also, the algorithm is also taking care of explaining well the data and explaining well the response. So on the same data set, we use the fasted glycemia as the outcome, and we compute the supervised analysis, the supervised model, which is the consensus OPLS with the R package, which was developed collaboratively by the team of Marie in Toulouse with Céline. I saw Céline also in the audience there, and the SIB, my colleague Vanu, which will talk later on with Céline, developed this package. And then with all the scores, as before, the variable loadings and VIP and the contribution of block, you can interpret your model. And with the loadings and VIP, you want to understand better how the variables are behaving and are able to predict your answer. So here, as we are in the orthogonal framework, we predict the response with the first component, which is called the predictive latent variable. The orthogonal latent variable here is summarizing the information that is not related to the outcome, but which belong to the data set. So as we ask the model to predict the fasting glycemia, the samples are well ordered according to their fasted glycemia level. Then we can see which variable are giving more weight, are more important to predict this fasting glycemia. And we also test the significance of the model doing permutation tests. So here is the results of our model, and you can see that it's really different from the histogram of all the permuted models. So, but now you have results of a statistical model, and you want the biological interpretation. So, how to put your the results of the model into the biological context? You can use the loadings and the VAP to interpret the contribution of variable to the model. You can visualize for example, the lipid influence to the fasting glycemia modeling across all the tissues here. And for the transcriptomic data, we did gene set enrichment analysis with the KEGG database for each tissue. And then we could compare which pathway were deregulated in which tissue. So as you saw just before, the next step after the data integration is to put the results into the biological context, and then you will need accurate databases to put stable identifiers on your molecules, on your genes, etc., and be able to then use metabolic networks, for example, integrating the different levels of the biology. If you need some more material, you can have a look at our GitHub, the training GitHub from the SIB, to help you put the results into the biological context. And then when you have new hypotheses, you have a lot of different pathways or molecules that were deregulated. You have to validate the obtained hypothesis. As you went from a large amount of data, you were acting like a funnel, and then you have a set of variables that seems to be important, and you will go again into the experimental part and try to validate those discoveries. So if you want to have additional course material, you can visit the GitHub of the course we gave with Julien Bocard and Vanu. Everything is freely available on the GitHub. You can also find the other SIB trainings that could be helpful for you. And with that, I will thank you all for your attention. And please, if you have questions, do not hesitate.
Thank you for your insightful presentation, Florence. Now we can move on to the questions, as I don't see any in the chat. So guys, if you have one question, don't hesitate, don't be shy. But in the meantime, I have some questions. So I was wondering that, in your sense, with your expertise, what is the most critical step in the multiple chemometric workflows that you've presented? What is the most critical step that you have emphasized?
I didn't speak about it, but I think it's the processing of the data, from the data coming just from the instrument to the statistical work. You really need to focus on applying the right normalization method, the cleaning, the annotation, and this is, if you want to have a soundful model, you need to really take care of this first step. I think it's really but as we all know, it's the hardest part, maybe,
yeah,
to really well prepare your data.
Yeah, we agree. Fair enough. So we have some questions in the chat. First question:
yes.
how to choose a method along the whole forest of methods?
I choose the method that I can understand. And so I've chosen those methods because they are really close to the, for example, PCA and OPLS. And so I can understand how they work. at what you need to take to pay attention to prepare your data. I can understand the results of the models and how to draw links between variable and sample, etc. So for me, this is important. If I understand the algorithm, if I understand the method and how the results are presented, it's a good way of choosing.
Thank you. Next question from Nuria. I was wondering which could be the best tool to perform low-level data fusion for repeated measures. How is that considered in the analysis?
I think for repeated measure, you can consider this maybe with other type of method in adding a third dimension to the data, but it may be difficult. You can also just consider the repeated measure maybe as a factor, but it's difficult to answer just like I cannot give you the name of one specific method without seeing the data, actually.
Next question from the first one to ask was, can you explain a bit more about the dimensionality reduction state that you applied before CCSWA on your paper from 2024?
Yes, it's the weight engine correlation network analysis. So it's a method that exists for a long time, and it's more or less it's clustering of variables. So the variable that are correlated, you compute correlation between all the variables, then you clusterize them and you cut at certain depth of the dendrogram, and then you have new modules. Those modules are summarized by the first component of the PCA of each module. And so it summarizes really well the data without losing information. It's WGCNA, the keyword, if you want to have a look.
Thank you. Another question, how to handle the fact that some variables are shared between blocks? Could you tell us how to put the adequate weight with the low-level fusion data?
I'm not sure I understand fully the question, because you mean the variable can be measured maybe in the different blocks?
I mean, if you have different blocks, but with variables that are shared in the different blocks. I mean, if you use different methods to measure the variable, but some are common to the different blocks. How do you handle?
Yeah, for example, you measure with a different LCMS method, and you can keep them in the different blocks because they are measured independently in the different blocks, and maybe you will gain insight because they will go in the same direction. The signal will be confirmed from the different blocks. But it's always a question, because, or maybe if it's, for example, two LCMS method, you know that one variable is better measured in one LCMS method. So I will choose the right method that measured the variable really accurately, to avoid going in different direction, because we know that for some metabolites only one mode is really accurate, and the other one we have suppression of ion or something like that.
Okay, thank you so much, Florence.
Thank you, Florence. Another question from Guillaume. I'm not completely sure how you can merge block of very different size, meaning mass spec metabolomics with enzymatic single dosage compound, for example. An extreme one here.
No, I also faced this quite soon, quite often, and those methods are well suited for that, because you compute the the square matrix first. So in the algorithm, then the blocks have the same dimension, which is the dimension of samples. So it's perfectly handled. I applied the first step with WGCNA also to visualize more easily the loadings, for example, but the model is really exactly the same with all the different variables. And I had the transcriptomic data and lipidomics data, so 15,000 of genes and only 200 of lipids, and it's okay. So with this first step in the algorithm, which where the matrix, the small matrices are computed, it enables to take care of this size discrepancy. And with the weight, the iterative weight computation, you will give the more weight to the block which has really more information, not more variable.
Okay, thank you, Florence. We have time for one last question. Sorry, others. So the last question will be from one of our chat here attendees. Marine asked, do you think that one day we will have a consensus PCA or?
It's called ComDim. It's the first method I showed, actually. It's maybe I didn't explain well, but the ComDim method is the unsupervised method. And so you can use it as a consensus PCA. It's not called like that because it was developed by another team or two other team. And so, but it's the descriptive method, the unsupervised method that is applicable.
Okay, so that concludes this first talk. Thank you again, Florence. Thank you for all the community. Thank you warmly.
Thanks a lot.
Thanks. And we will now give the mic to the following speakers. Find it. There we go. So we'll now give the mic to Tran, a senior computational biologist that Florence talked about earlier, also from the Swiss Institute of Bioinformatics in Lausanne, and he will present his work on multi-omics data analysis applied to diabetic kidney disease. Manu, the floor is yours. Oh, you're muted, Manu.
Wait, can you hear me?
There you go.
Thanks. Yeah, thank you. Thank you for having me. It's my first time here in the community. Despite I've heard about your community thanks to Florence. And so yeah, I'll present you a case study on the multi-omics data analysis in the framework of diabetic kidney disease. So you might have known very well about insulin. Insulin is a peptide hormone produced in the pancreas and is encoded by the insulin gene in human. And there are evidence about insulin resistance increasing the risk of DKD, diabetic kidney disease. And kidney cells will become insulin resistant in a diabetic environment. So in this work, in this paper that we published a couple of months ago with a BITKD, that is IMI project, BITKD consortium, that where we explore the molecular changes in insulin-resistant human kidney cell models. So here we have transcriptomics and proteomics in 4 cell types. Let me just take the 4 cell types in kidney. So here we have the kidney, we have 4 cell types, glomerular endothelial cells in red, podocytes, mesenteric cells, and proximal tubular cells. We have 2 conditions, the basal. and the one where we mimic the diabetic environment. So we input some high glucose and high insulin with some TNF alpha IL-6 to mimic the environment. And also we have the two conditions with the overexpression of the insulin receptor. Okay. So in summary, we have 4 cell types, 2 treatments, basal and diabetic environment, with or without insulin receptor, and we have 5 replications for each of them. So this one is the overall picture with a PCA blood where we see a variation, a dominant variation between the cell types, comparing to the variation between the two conditions where we expect to see the variation is between basal and insulin resistant. Okay, so that's why we try to analyze the difference between these two conditions in each cell type separately, and later on we decide to use only the with insulin receptor where we have kind of reinforce of the signal between the basal and the diabetic environment. Okay. So in each of them, in each cell type separately, we perform a differential analysis where we have genes and proteins here on the right side. This one is a volcano plot. You see in a full change, I mean the level of change between the two conditions, and the FDR kind of p-value, corrected p-values that show the significance of the change. So the proteins and RNAs, they change differently between the two conditions, and that's what we want to investigate later with the pathway analysis. So we have only one contrast on the 4 cell types with the 2 omics. So we perform the GSEA as Florence told you before with GAIC. So now here we perform on Go, the gene ontology. We have the molecular profile about a fold change, p-values, etc. We have a gene set database. Gene set is a set of genes annotating to some function. For example, you have agorastol biosynthesis with 10 genes, for example. And this method, we need a ranking of genes that we can use the folding, the fold change, or the signal to noise, or the p-values, etc. So we need a ranking, and we perform random work along the ranking. And if we meet one of the genes in our gene set, we move up. If not, we move down. So in such a way, we define a curve like that, and we have a surface of this curve that defines the enrichment score. Okay. So it's very classic that we have a table of gene sets. For example, the first one, we have the gene set of this Go term that has 7 genes with an enrichment score of about 2 and a p-value, etc. And at the end, we have a table for transcriptomics. We have a table for multi-omics that we obtain separately. So now we try to put the 2 omics on the same table. We put 2 persons in the same room to see how they influence each other, how to the final decision. That's where we use the consensus of BLS to integrate transcriptomics and multi-omics. So this method was developed by Julien Boucard from University of Geneva, and we redeveloped in R as an R package with new functionalities by improving the computing time, bringing on the capacity to deal with multiple classes, different kernels, kind of nonlinear kernels, and also bring on the capacity to predict the new samples to provide the interpretation of the model as well. So these methods, we have multiple blocks. You have multiple blocks, X1, X2, Xk, with the same observations, with different numbers of features, and we have one response. Okay, this response can be continuous, can be binary or multiple classes, and that's the interface of the method. The method will take into the data of multi-blocks, the response, the number of predictive components, etc. It will provide you a number of functions to visualize the result. For example, the two main informations here is about the score and the loadings. Okay, the score, you have, for example, here in this model, we have the response, the binary response between the red and blue. So the first component here will try to separate the best, the variation, the two classes. Okay, and the loadings is the contribution of the variables of genes or proteins to the variation between the two classes in this dimension, the predictive, the P1 here. So with this contribution, we also have a ranking of genes or proteins. So with that, we can again perform a GSEA. So now we have kind of a lot of GSEA results. We have two GSEA results from the individual differential analysis on genes and on proteins. We have two from genes and proteins from the multi-block, the consensus OBOS. So we have four tables. So the goal now is to find a consensus of the four results and then to summarize the goal terms together using the semantic similarity between the goal terms. This is so here, for example, we have the four tables of goal terms together in a huge plot like that, and we have in red the up-regulated pathway, and in blue the down-regulated pathway. Okay, and the pathways they are grouped together into the same branch inside the gene ontology. It means that those in the same color, usually they belong to the same function, same biological process or molecular function, etc. So for example, here, if I took one block with the red everywhere, so it corresponds to the immune pathways among the four cell types. We had 4 cell types with the same response, with the same phenotype of upregulation of these immune pathways. In another case, we can see this mitochondrial dynamics is upregulated in one cell type, but downregulated in the other 3 cell types. So that's kind of signal, that's kind of results that we expect to see the common or the cell type specific pathways, and thanks to the consensus between the individual analysis and the multi-block analysis. So here, for example, the main finding in this paper is that we look at the goal term, but not only goal term, we can use also the gene set annotation like reactome, where we have a relation between the goal terms. For example, GEC, we don't really have the relation between the GEC terms, but here we have the goal terms, reactome, that we have a tree, we have a DAG. that summarize the relation between the gene sets. So for both of them, we still have kind of very disruption, a traumatic disruption of the pathways that governs the mitochondrial dynamics. So we have in this cell type, the endothelial cell is upregulated, but down in the other ones. But we can only observe that in proteins, but not in transcriptomics. So the message here is that we need to use multi-block data equation along with the individual omics analysis to put them together, performing the pathway enrichment in different types, the otome, the cake, the reactome, etc. And the final goal is to find the consensus between all of them. So that would provide a significant result at the end. Yeah, with that, we'd like to thank our co-authors in the BJKD project for the paper, and thank you for your attention.
Thank you again, Vandu. We know for your remarkable presentation. And let's move to the question. I did not see some question on the chat, but maybe we will have in a few minutes. First of all, I would like to ask if you, I saw that you presented some data sets with proteomic and transcriptomic data set.
Yeah.
Have you ever used these methods with metabolomics? Do you think that using transcriptomic and metabolomics could be easier to analyze or better to make some good correlation between these kind of data sets? What do you think about that?
I would say it depends on the quality of the metabolomics data. What I have seen in one project is that the quality of metabolomics data is far away from that of transcriptomics. So equating them was not helpful. Just like you put two persons in the same room, and one is very rational and the other one is just there to give random decision. So it doesn't make sense. But if we have very good data, I know that Florence she showed me very good data. So, but if we have data in the project, so it can be absolutely helpful.
Okay, thank you. No question in the chat. Maybe I know that you have to leave also at 4 because you have some stuff to do. So thank you so much to give this talk today. And now we can maybe move to the next presentation.
Yeah.
Okay.
Thank you again, Manu. And we now welcome the next speaker. So yeah, we now welcome our final Swiss speaker of this webinar, Josefina Lascano Maillard, also from the SIP, from the Swiss Institute of Bioinformatics from Lausanne. She's a computational biologist, and without further ado, she will present her work on the multiomics models in a MASH-related fibrosis. So Josefina, the floor is yours.
Thank you for the kind presentation and for the opportunity to talk here today. I wanted to show you an example study where we try to model MASH-related fibrosis pathobiology via multi-block integration. So first of all, what is that we are trying to model? MASH stands for metabolic dysfunction associated steatohepatitis, and it arises when liver starts to accumulate fat, develop inflammation and ballooning, and eventually it can also lead to fibrosis development and cirrhosis at later stages. It's quite, the prevalence is quite high in the population, and the problem is that there are still few clinically relevant biomarkers. The aim of the QuidNASH research project consortium is to move from a liver biopsy that is invasive to a virtual one, and also to increase our knowledge about the MASH pathophysiology. So in this study, we focus on a clinical cohort and a mini pig model, which was shown to recapitulate quite well the main phenotypes from MASH. And in both populations show varying degrees of MASH and also fibrosis stages. We aim to integrate transcriptomics and metabolomics data with laboratory measures, and additionally for mini pig, lipidomics profiles were also measured, and so we integrate those as well. But as you can see by the different numbers here, we have quite a lot of differences between dataset sizes, and also we expect some redundancy at some extent between the data sets, for example, between metabolomics and lipidomics data, but also within a single data set. So we would like to focus on common variation that is related to the phenotype, and therefore we would need to reduce dimensionality and also summarize the common variants in a biologically meaningful way. So we therefore adopt the following strategy prior to building our multi-block model. We perform weighted gene correlation network analysis, WGCNA, in order to reduce the dimensionality of data, and we run a supervised model with consensus OPLS in order to identify transcripts, metabolites, lipids, and biochemical measures that are associated with fibrosis score. Now, if we take a look at the common variation related to fibrosis, we see that the four data types contribute to the model in a complementary way by looking at their colors that are assigned to the different data sets, and the VIP values. VIP stands for variable importance projection. That can be negative or positive depending on whether they are low or high in fibrosis. The model is significant, as assessed by permutations, and human and mini pig samples are distributed gradually in the predictive component space with increasing fibrosis. And so now we can dig on what is the biological meaning that is associated with this common variation related to fibrosis. And for this, we focus on the 10 top VIP modules, so the 10 first modules with the higher VIP values. And for each gene and each metabolite, we can have measurements to relate it to one of the top 10 VIP modules. We then, in this way, we obtain a ranked list of genes and metabolites and perform gene set enrichment analysis for both of them. For metabolites, it would be called a metabolite set enrichment analysis In a second step, we combine the p-values of the rich pathways in order to select a shared and common pathway between species. As a sanity check, we look at known processes that were already described in MASH and fibrosis development are present, and indeed they are shared by human and mini pig, like is the case for glycine, serine, and threonine metabolism or primary bile acid biosynthesis. But also some metabolic pathways are specific to some species, as is the case for mitochondrial electron transport chain, histidine metabolism, or cellular responses to stress. So now, within these pathways that we identified, how do metabolites and genes interact in the context of fibrosis? We can use Bayesian networks to test hypotheses and also assess the likelihood of causal relationships. For example, if we want to assess what is the probability that someone has a headache, knowing that this person sleeps less than 6 hours, and how this will be updated if we knew that this person feels fatigue or was infected by a virus. So we can also learn the structure of the data from scratch. But two main limitations from this method are that we often work with very small sample sizes compared to the population we want to describe. And often we don't know if all possible causes were measured, actually. So we need to add knowledge to these networks, and therefore we decide to focus on interspecies shared components, and also back up our results with the literature. We use Bayesian Learn, our package, to learn the structure of gene and metabolites expression data. We learn the structure with hill climbing, search algorithm, and Bayesian information criterion score for Gaussian data. And we focus on those components which have causal relationships involving fibrosis that are shared between the species. And here I'm showing only an example to you of components that were shared within the phase 2 conjugation of compounds pathway. These components were shared between a human and mini pig, including the causal relationship between OPLA and fibrosis in yellow. But we can see that is detected in 2 Bayesian networks of human and mini pig. We see also with in green and orange other relationships between genes that are conserved between species. We also confirm this at the expression level, that OPLA is affected during fibrosis development, and this is true across human, mini pig, and mouse, because we also checked in another data set that we had from mouse. We see also these the same relationship, causal relationship between OPLA and fibrosis, by the way, in this data set. Furthermore, in the literature, we also find evidence of what we observed, where a model of OPLA knockout, a mouse model of OPLA knockout, leads to fibrosis in the heart and in the kidney, but they don't have direct measurements of liver fibrosis. So the take-home message here and the future avenues are that mini pig, it's an animal model that has a good translational value, that we can see because of the shared mechanisms with human. Thanks to Bayesian networks, we can also we could identify candidate targets for a future fibrosis treatment research. We could, in the future, it would be interesting to characterize further the OPLA knockout model. in the context of MASH and liver fibrosis. And potentially we could make available results to the research community in the form of a resource. I want to thank my colleagues at SIB and all our collaborators at the QuitNASH Consortium. And thank you for your attention.
Thank you very much, Josefina. The presentation was very clear, and thank you. Does anyone have any question? Let's go to the chat. No question for the moment. So maybe I can ask you one to let people write in the chat. So I don't, maybe I missed something during your talk, but I would like to know if you know the role that OPLA plays in the liver. I don't see the name of these genes and what is
yes, sorry for that. I didn't detail the name. It's actually a detoxifying enzyme, but we didn't do any try to validate in the laboratory for the moment, but we have these evidence that come from previous studies, and we are trying to see if we understand how this can lead to fibrosis development, but it seems to be something to see with the cytotoxicity of lipids in the liver, and this enzyme it's protecting the liver from this cytotoxicity. But and in the case where it's decreasing in expression, fibrosis it's can be
increasing.
Yes.
Okay.
Thank you.
And finally, the final idea of this project is to use either the metabolites or the gene expression of OPLA, the transcript of OPLA to better diagnose the NASH or not.
Yes, at long, exactly. Yeah, it's a long-term goal, but we were happy to see that is not the only causal relationship we found, and it's always happening within these pathways that were identified really in a data-driven manner. And I think it can guide also future research, people that would like to look at different test particular hypotheses with these causal effectors. Yes.
Okay, thank you again so much, Josefina, for your talk. And now maybe we will move on the next presentation.
Thank you.
Yes, thank you once again, Josefina. So now we move to the final speaker of this webinar. Find him. There you go. So Tito Damiani is a postdoctoral fellow at the Institute of Organic Chemistry and Biochemistry of the Czech Academy of Sciences in Prague. Tito works on the biochemistry of plant-specialized metabolites in the renowned Tomáš Tuškal group, and we present PipeNet, so a tool for multi-omics approaches. Tito, the floor is yours.
Can you hear me?
Yes.
Good. And now you can see my slides, right?
Yeah, perfectly fine.
And you can also see my laser pointer.
Yes, we can see the project.
Perfect.
Yeah.
Thank you. Good afternoon, and thanks for the introduction and also for the invitation to this webinar. And so today I'll show you some, like, the way we're trying to do multi-omics in our lab, which might be a little different from the examples we saw so far, and also like some preliminary results from my main postdoc project, where we're trying essentially to combine metabolomics and transcriptomics data on, let's say, plant family scale for the elucidation of the biosynthetic pathway of piperazine alkaloids. So in our lab, we work on plants, and The reason why we're interested in plants is because plants are fantastic chemists. They produce a stunning diversity of secondary metabolites, and many of these metabolites have found applications in our everyday life, including applications as pharmaceuticals. For example, in this slide, you might recognize paclitaxel, which is a very successful anti-cancer drug, or artemisinin, which is a used for the treatment of malaria, and its discovery was awarded the Nobel Prize in 2015. And so one very active research area in our lab is indeed the discovery of new plant natural products that could one day become a successful drug or find any other application relevant for our society. But another thing we're really interested in our lab is also to understand how the plant is able to biosynthesize these compounds, which means to figure out what are the biosynthetic genes and enzymes involved in the biosynthesis in the plant of these molecules. Because after we know that, then we can fairly easily plug the pathway into chassis organisms like microbes or yeast, and guarantee a more sustainable production of these phytochemicals without the need of sourcing the molecule from the native plant in the wild, for example. Now, the problem is that this step of finding the biosynthetic genes in plants, especially non-budded plants, it is really hard because plants have very big and complex genomes. So finding those few 4, 5, up to 10 genes may be necessary in the plant for the biosynthesis of this compound. It really becomes like finding a needle in a haystack. And so another thing we do in the lab is to develop workflows, mainly computationally based workflows, to make this process faster and more efficient. And that is the main topic of my main postdoc project, where I'm using this plant family as a model case to develop this approach. And the plant family I'm talking about is this Piperaceae plant family, which is a big plant family with over 3,500 different species distributed all around the world, from all around the world. And it's widely recognized as a rich source of bioactive natural products, alkaloids in particular. More than 300 different alkaloids have been already isolated from this plant family and have shown a wide range of biological effects on humans, from analgesic to anti-cancer properties. And so let's say we wanted to find some potentially interesting new compounds. First thing we needed to start off was, of course, some samples, and these are tropical plants. I'm based in the Czech Republic. These plants don't really grow very well here, so we had to connect with several botanical gardens around the country, and we managed to collect 13 different species of plants from this family, and we confirmed their taxonomy using DNA barcoding. And then from each species, we collected 3 to 4 tissues, leaf, stem, root, and fruits when available, in triplicates. And the next thing we did was to profile the metabolic content of all these samples using untargeted metabolomics. And this is a schematic representation of the workflow we normally use to process, to analyze our untargeted metabolomics data. So after very briefly after the data acquisition, we used the MZ-Mind software for what is called the feature detection step. So just turning the complex raw spectral data into a list of detected metabolites and their abundances across the different samples. Then we use molecular networking, which is a data analysis technique for metabolomics data that groups together unknown metabolites that supposedly have a similar chemical structure because they have similar fragmentation mass spectra. And then in parallel, we also use machine learning-based tools for the prediction of the chemical structures of this unknown metabolites. So then we can overlay these predictions on top of the network, make it more informative and easier to explore. So what you get out of this is something like this. This is the molecular network that resulted from the processing of my entire data set. You can see that it's quite massive, so 14,000 different nodes that here you can think of them like individual metabolites detected, organized in almost 4,000 molecular families. And what was nice is that when we overlaid the compound class predictions from this software, more than 1,000 nodes in the network were predicted to be alkaloids. And actually, many of them grouped together into big molecular clusters. And some of them, which might represent, like structure-related molecules, alkaloids that belong to the same biosynthetic branch. And so there were like a few networks that drew our attention, in particular these two big ones predicted as piperidine alkaloids, meaning that they're supposed to contain the piperidine ring in their chemical structure. And these plants are famous for the production of this class of alkaloids. And this other one that was predicted as isoquinoline alkaloid, meaning that they are supposed to contain this core scaffold in their chemical structures. And this was already quite cool because there was no report of this class of alkaloids in this plant family. And so we look into more details into these networks. And for the piperidine alkaloid network, we found several both reported and unreported piperidine alkaloids that mainly fell into two main biosynthetic branches, one leading to piperlongumine, so this guy up here, and piperlongumine-like compounds, and another one leading to piperin and piperin-like compounds. And so things started to get interesting when we look at the abundances of the individual metabolites across the different species that we analyzed that are now shown with these pie charts. And we started to see some patterns. For example, the blue species only show up in the piperlongumine branch, whereas the yellow, the light blue species only show up in the piperin branch. And same story for the isoquinoline alkaloid network, where most of the compounds from this pathway, where we found like several members of this alkaloid class, and many of them are present in all the analyzed species, whereas others are very specific to one, like these two guys here, are very specific to one single species out of the 13 species we analyzed from the same family. And so now the question we're trying to answer is whether having this information about the interspecies, or let's call it like taxonomic distribution information of metabolites, can help us in finding the corresponding group of biosynthetic genes we're interested in. And so we're trying to switch from what is like the standard approach in this field, where you normally pick one target molecule you're interested in, and then you study its pathway in one single native plant species, to what we call a more network approach, where we try to consider the whole cluster, the whole class of these alkaloids, and how they distribute across the different species within the same family. Because, for example, if we observe the same alkaloid to be present in multiple species, let's say 3 species, in the same species, we can expect the same biosynthetic genes to be present. And so maybe we can map this common biosynthetic steps across the species and use this information to prioritize the genes to be then, the genes that follow the same taxonomic distribution that then will need to be tested experimentally in the lab. And so to test this hypothesis, to see whether we are able to do this and use this information in a meaningful way, we're building this Multiomics platform where, as I just showed you, we take a set of different species from the same family, collect different tissues, and then use untargeted metabolomics and molecular networking to, let's say, reconstruct the biosynthetic branches related to to alkaloids and map their distribution across the different species and tissues. And then in parallel, we do a very similar thing for the genes using RNA sequencing and de novo transcriptomics. Now, if you're not familiar with these technologies, they're conceptually very similar. Like there is a first stage of untargeted high throughput analysis for the RNA sequencing as well. And then there are bioinformatics tools that can be used to align, let's say, genes detected in different species, but they're most likely the same. They most likely perform the same function in the different species. So, like, long story short, at this point, we're able to say, okay, I observe this metabolites in this species, but not in this species. And we're able to do the same also for all the genes that we can detect with our sequencing. And so what we would like to have at this point is a way to automatically connect each alkaloid branch to the corresponding group of biosynthetic genes in a data-driven manner. And as you can tell from the numbers on the slide, we're dealing with quite massive data sets, like 14,000 different nodes, 4,000 networks almost for the metabolomics data, and almost like 1.5 million genes for the RNA sequencing data. And so we were expecting like a lot of connections and correlations that were like just noise. And so what we needed was a scoring algorithm to kind of distinguish the true connections from the random ones and at least rank them higher and prioritize them for the experimental testing. And so once we got this set up, the only thing that was left to do was to just try it out. And we started from the isoquinoline alkaloids network, and specifically from the very first step in the pathway, which is the coupling of this dopamine with this aldehyde to form norcochlorine, which is the first precursor of this class of alkaloids. And this reaction is known to be catalyzed by an enzyme called norcochlorine synthase. And so what we did was to take all our candidate genes for this reaction. So you can see there are like quite a few. And we run our algorithm, and those that you see here in as bars are the corresponding score given by the algorithm to each candidate. And to test whether these numbers actually make any sense, we picked four of these candidates, and then we tested their activity. So we expressed the genes in a different organism, and we checked whether it would produce the expected compound. And to be honest, to our it went a little bit beyond our expectations. 3 out of 4 of these genes actually showed activity, and the only one that didn't show any activity is indeed the one scored the lowest out of the ones we tested. So it seems the approach makes sense, and it's working. So now we're gonna continue with the downstream steps in the pathway and see how it goes. I expect some class of enzymes to be more challenging, like acetylcholine P50s, CoA ligases, and stuff like that. But so far, it seems quite promising. Okay, so to sum this up, we're developing a multi-omics platform to connect alkaloid biosynthetic branches to the corresponding group of biosynthetic genes in a data-driven fashion. We're going to use this platform to elucidate the biosynthetic network of piperidine and isoquinoline alkaloids. And although this approach is developed on the Piperaceae plant family, in principle, it can be generally applicable to other plant families of interest. And so with this, I'd like to conclude, and thank you all for the attention, and again for the invitation.
Thank you so much, Tito. It was an awesome work. So, okay.
It's in progress. It's like
Yes, but there is a lot of work, I think, to do that, to acquire this data set and to look for that correlation. And so thank you very much. We have a question in the chat. In a practical way, how do you combine the alkaloid synthetic branches with the transcriptomics orthogroup?
So I guess the question referred to the scoring algorithm. Like, long story short, we take into account different layers of correlation. Like, one is the, maybe, can I go back to the slide?
Yes, for sure.
So, so one layer is how well the distribution across species of the entire biosynthetic branch, not the individual compound, of the entire network of alkaloids, how well it correlates in terms of like species distribution with the group of biosynthetic genes, and also how well their abundances and expression correlate across the tissues. And then we also integrate some other parameters like gene annotation, metabolism annotation for the refinement. But one thing that I would like to highlight without going too much into details is that we are not trying to correlate one gene to one single gene to one single metabolite, because I'm afraid that wouldn't work in this, at least in this application. So we're trying to correlate and score the correlations between the entire biosynthetic branch and groups of genes aligned, let's say, across different species.
Okay. Thank you so much. Again, Tito. I don't see other questions, so maybe you can thank you again for your talk today. Thank you so much. And now I let the mic to Thomas to conclude.
Yes, thank you. So I'm going to share my screen quickly. There we go. So yeah, once again, thank you for your very educational and clear presentation, Tito. A big thank to our four speakers and to the audience. As I said earlier, the recording of this webinar will be available soon on the RFMF platform for the replay. And just before you leave, I have an announcement.
Big announcement.
A big announcement for the upcoming event. So we'd like to give a quick reminder about the upcoming RFMF Scientific Days. So they will take place in Paris from the 13th to the 16th of June this year at the Université Paris-Cité. So all the information is available via this QR code on the right of the slide or through the ScienceConf site page by typing 17 JS RFMF. So the deadlines for early bird registration and oral presentation is on the 28th of March, and the deadline for poster submission is the 25th of April. So you still have some time left. And finally, we'd like also to emphasize that travel grants of up to 500 euros are available for early career researchers to attend the conference. And the application conditions and information form can be accessed through this via this QR code or through the website of the RFMF page in the Bourse de Voyage section.
And please submit before the beginning of April, because we have 2 months. You have to apply 2 months before the first day of the conference. So please, if you want to be selected for these travel grants, consider applying before the beginning of April.